The nature-nurture debate is one of the oldest unanswered questions in science. How much do our genes determine our risk for disease compared to environmental factors such as lifestyle or diet? Today, I'm excited to share some new findings which may answer this fundamental question. Hi, my name's Tony Monaco. Many of you may know me as the president of Tufts University, but I'm also a geneticist. I have been fascinated with the nature-nurture debate since the early 1980s, when I helped identify some of the first genes for human rare inherited disorders like Duchenne muscular dystrophy. Finding genes for rare disorders was also a path to finding diagnosis and treatment. However, for muscular dystrophy, this took 30 years, from the first gene discovery in 1986 until the first FDA-approved treatment in 2016. These early discoveries inspired the Human Genome Project, which was an effort to sequence our DNA as a blueprint for life and heredity. At the turn of the century, we celebrated the completion of the Human Genome Project. Scientists and clinicians used the DNA sequence to identify most of the genes that cause rare inherited disorders. However, when similar approaches using the DNA sequence were used to map the genetic effect in common diseases such as heart disease, diabetes, or mental health disorders like schizophrenia or depression, only 40% of the genetic effect actually mapped back to the human genome. This apparent paradox is called the missing heritability of common disease. The missing heritability paradox raises the following questions. What is heredity if DNA only contributes 40% of the genetic effect in common disease? Is there another system not based on DNA which can provide information to the next generation? And could a non-DNA-based system provide information to children about environmental exposures in parents? Answers to these questions have come from model organisms like yeast, flies, worms, and mice, where environmental stresses such as food scarcity, general stress, or severe infection in parents contributes directly to the health and behavior of their offspring over several generations. In each case, the sister molecule to DNA, called RNA, was directly implicated in what is called an epigenetic effect from environmental stress. Many scientists have studied how DNA is modified or decorated as possible epigenetic effects. But these DNA modifications are erased in sperm and eggs and therefore are not heritable. In contrast, RNA is directly heritable like DNA from parents to children, and they may work together as the heritable material. So how does this fit into our current concepts and understanding of how heredity works? Let's start by deconstructing our current model about how DNA contributes to life and heredity, called the central dogma. The central dogma is taught in every introductory biology course across the world. It states that DNA has two main roles as the blueprint for life and heredity. One, it can make copies of its genes as RNA, which provide many of the functions in the organism, such as the proteins which make our hearts beat and our brains think. Second, DNA can also make copies of itself to generate the heritable material that's passed from parents to offspring as a blueprint for life. Now, let's reconstruct this model by inserting RNA as a second possible heritable material alongside DNA. RNA can change its levels and its genetic code in response to environmental stress, and some of these changes are heritable. We learned from the COVID-19 pandemic how rapidly RNA can evolve and mutate in viruses and cause severe, long-lasting disease. So viruses can be placed alongside other environmental factors and exposures in parents, such as diet, stress, bacterial infections, and substance misuse. In this reconstructed model, DNA and RNA are both contributing to heredity, but the environment works mostly through changes in RNA. I was inspired to look at RNA, at regulating the potential cause in the rise in mental health disorders, which can't really be explained simply by changes in DNA. This exploration was validated by the animal models and the sheer fact that the RNA is the only molecule which can rival DNA for the amount of information it can store in its genetic code. So in addition to storing information, RNA molecules can find partners through a joining process called splicing. The joining process acts like a memory, since RNA can find older evolutionary partners amongst other RNAs in the cell that were previously established to fight off a viral infection. Given the need to respond to rapidly evolving viruses, RNA can also join into new partnerships, which may prove useful to the organism to fight off a newly encountered viral infection. These new RNA relationships, if beneficial over many generations, will eventually drive changes at the DNA level, and therefore RNA and DNA can co-evolve. RNA has the main role of adapting the organism to environmental stress and remembering those old and new partnerships to improve the response upon repeated exposure. Other environmental factors which have been shown to change RNA levels are bad diet, substances of misuse, bacterial infections, and stress. And some of these RNA changes can be passed across generations, influencing health and behavior. Our team at Tufts have found that through splicing with partners, both old and new, RNA can produce a circular form, which is stable and has been found in every living organism on Earth. Circular RNAs are produced when a linear molecule joins their ends together through a splicing event. It forms a circle instead of joining with another linear RNA molecule. It's a backsplice in forming a circle with its own ends. And these circular RNAs are very stable because the closed circular structure makes them resistant to degradation. They seem to be important in fetal development, especially in the brain and heart, and can be edited, thereby providing further flexibility in their genetic code to match environmental challenges. In the last decade, circular RNAs have been identified for many human genes, but they're usually smaller than the genes itself. What our team at Tufts found was very large circular RNAs, which we call jumbo circles, as a nod to their size, as well as our beloved mascot, Jumbo, here at Tufts. The jumbo circles encompass many functionally related genes, and therefore provide a coordinated response to maximize efficient reactions to environmental change. One set of jumbo circles seems to be involved and produced after environmental stress caused by viral infection. The genes on these jumbo circles help the organism return to normal after the attack, in a cleanup process of sorts, to get the trains back running on time. Without jumbo circles, this cleanup process after stress or infection may not work properly, and after repeated exposures, could increase risk for mental health disorders and neurodegenerative diseases. The ability to form these jumbo circles is heritable and would be part of the RNA memory of previous environmental exposures to the next generation. Our team found a dramatic example of jumbo circle formation in turtles and tortoises, which are known for their long lives and negligible aging process. They are extremely good at making jumbo circles with tens of copies of the RNA element needed to join the ends together. Perhaps this spectacular jumbo circle making ability helps them overcome the stress of infection, and therefore they don't ever accumulate the cellular garbage which can lead to aging, and in humans is the cause of neurodegenerative diseases like Parkinson's and Alzheimer's. Small and large RNAs may also help us understand mental health disorders to improve diagnosis and treatment. Mental health disorders are some of the most heritable of the common disorders, yet DNA changes that have been found only contribute a small part of that risk. Therefore, there's plenty of room for a parallel RNA effect working alongside DNA. It's been shown in the brains of individuals and children with neurodevelopmental disorders that they have a reduction in the editing of their RNA. And we're exploring whether that RNA editing reduction may be because they have a reduction in circular RNAs. This could have direct diagnostic and treatment potential, especially given that the proposed RNA mechanisms are transient and therefore reversible, unlike DNA changes. This RNA mechanism may have been developed very early in evolution in response to RNA virus infection in water-dwelling animals such as clams, mussels, oysters, and crabs. It seems we still have this ancient antiviral mechanism in humans, where similar RNA partnerships are used to combat virus infection. Understanding how our RNA can fight and remember viral infection by joining other RNAs to form jumbo circles should provide an alternative route for antiviral treatments. The virus that causes COVID-19 is an RNA virus, so boosting our natural antiviral detection system should provide therapeutic potential and help reduce severe illness and hospitalizations. This project has been a journey of scientific discovery where many faculty, staff, and students across Tufts University have helped me understand its significance to biology, evolution, and disease. I have revised the central dogma to include both RNA and DNA as the possible heritable material. The inheritance of RNA as a barometer of environmental change could revolutionize our understanding of mental health disorders, which are continuing to rise in this generation without an adequate explanation. Current treatments for rare inherited disorders like muscular dystrophy have successfully targeted RNA. Targeting RNA may lead to better treatments of common disorders as well. Perhaps one day, humans can live as long as turtles and tortoises if we can boost our jumbo circle making ability to overcome chronic inflammation from infections and slow down the aging process. Thank you.